This slide gives an overview of BCL2 conservation across metazoans, showing an evolutionary tree, protein sequence homology, and conserved structural domains. The central image highlights the BH3 binding groove and associated motifs that underpin both pro-survival and pro-apoptotic roles, while side panels point to cellular homeostasis and possible therapeutic targeting based on these conserved features. Here we examine lineages that lack BCL2, like ctenophores and certain nematodes, and what that means for apoptosis regulation. The diagram suggests complete family loss in some groups, yet maintain viability, indicating alternate death pathways. This absence reveals evolutionary flexibility. BCL2 regulated apoptosis is advantageous in complex organisms, but not universally essential for metazoan survival. This figure superimposes alpha helical bundles from sponge, trichoplax, and hydra BCL2 proteins onto human BCLXL to show a deeply conserved BH3 binding groove. That conserved hydrophobic groove supports both pro-survival and pro-apoptotic interactions and traces an evolutionary trajectory over 600 million years, suggesting this structural interface is a robust target for therapeutics that modulate binding across species. The slide outlines a fixed BH4, BH3, BH1, BH2 motif arrangement with invariant landmarks such as a conserved leucine in BH3 and an exon junction at GGW of BH2. These features provide reliable anchors for identifying BCL2 orthologs in newly sequenced genomes and enable robust genomic applications, improving orthology assignment by leveraging conserved structural motifs rather than relying on variable primary sequence alone. This panel focuses on the C-terminal transmembrane helix as an obligate localization and functional element, showing rapid sequence divergence but conserved structural presence. The illustrations link membrane targeting to mitochondria, ER, and Golgi, and emphasize dual roles, enabling back pore assembly for apoptosis and supporting BCLXL pro-survival activity, making membrane anchoring crucial for both pore formation and pro-survival functions. We trace the evolutionary appearance and conservation of BH3-only proteins, highlighting an amphipathic BH3 helix required for groove engagement. The slide suggests BH3-only motifs appear later in certain metazoan lineages with low primary sequence conservation yet retained functional necessity, implying repeated independent emergence and convergent evolution that fulfills lineage-specific demands for apoptotic regulation. This slide describes how large DNA viruses encode BCL2 homologs that mimic the canonical fold to bind host BH3 motifs and suppress apoptosis. Structural mimicry validates the groove interface despite minimal sequence similarity, allowing viral proteins to block host death pathways and promote cell survival during infection, which underscores the functional indispensability of the BH3 binding groove across diverse biological contexts. Here we focus on viral BCL2 proteins and their mitochondrial localization mechanisms, showing both C-terminal anchors and alternative N-terminal signals that achieve membrane targeting. The central message is that localization to mitochondrial membranes is a conserved functional trait necessary for sustaining host cell survival during infection, and that understanding these targeting modes could inform antiviral strategies exploiting this conserved vulnerability. This slide links BCL2 paralog diversity in early metazoans with the later emergence of adaptive immunity, suggesting an evolutionary trajectory of functional streamlining. Early lineages show many paralogs and high diversity, while vertebrates underwent selective contraction and specialization, correlating paralog loss with the need for refined checkpoints and specialized functions in more complex immune systems. The figure highlights mechanistic plasticity in apoptosis, showing arthropods and nematodes often lack canonical BCL2 repertoires yet execute caspase-dependent cell death. Without mitochondrial BCL2 control, these organisms rely on alternative routes, including distant BH3-only proteins that directly activate caspases, demonstrating that metazoan cell death networks can flexibly shift between BCL2-dependent and BCL2-independent mechanisms. This slide emphasizes the deep conservation of the BH3 binding groove from sponges to humans and the therapeutic potential that creates. By combining comparative structural models of basal metazoan BCL2 and human BCL2 with specificity screens, researchers can identify selective small molecule BH3 mimetics, leveraging basal structures to improve specificity and potentially open new avenues for targeted cancer therapies. The final slide frames BCL2 controlled mitochondrial apoptosis as an ancient core process that can nonetheless be repeatedly dispensable in certain lineages, affecting phenotype interpretation and therapy. An ancestral regulated pore forming mechanism persists, but lineage specific modifications or pathway loss create alternative survival strategies, which complicates knockout interpretations and points to evolutionary blind spots that could reveal therapeutic vulnerabilities.